Okay, welcome back. We are looking at properties of enzymes. Properties of enzymes. We've talked about them. Highly specific. And we've said that enzymes bind to specific reactants. They bind to specific substrates and catalyzed a single reaction or a set of reactions. Enzymes are also efficient and hence limit unwanted products, number of unwanted products. I gave an example of on the specificity, being highly specific. Give an example of digestive enzymes. For example, those that act on proteins. For example, trypsin, which is a digestive enzyme doing digestion. So it will only catalyze or cleave the peptide bonds on the enzyme, on the amino acids. We said that activities are affected by various factors, for example, pH, temperature, and other factors that we're going to look at later. Then we said that
my puff has brought electricity. Photo of it, please. Okay, Pop.
Okay, so we are saying that they typically help change one form of energy into another, a more usable form. And we talked about photosynthesis. We've talked about production of ATP. And then we are saying now that they typically do not act alone. And we are going to further elaborate on these core factors. Then should be the last part that we're going to do on Saturday. So they require helper molecules. So for example, core factors, which are helper molecules that are needed for enzymes to be able to function properly. The enzymes to be able to function properly. Then there are also the core enzymes as well. So these could be, these are numerous enzymes, these are numerous molecules or molecular substances, organic molecules, which are required by enzymes, certain enzymes. So this is a core, so these are core enzymes, core enzyme. And then there are also prosthetic groups. There are also what are called prosthetic groups. Prosthetic groups, which are also important. So usually the prosthetic group will signify a covalently bound core enzyme, covalently bound core enzyme. However, we're going to talk more about this when we meet next. So enzymes typically will require these helper molecules, the core enzymes, the core factors, and also we find certain enzymes, certain molecules are going to contain prosthetic groups. And then, very important, especially with regards to what we looked at in the preamble, when we talked about the reaction. Remember that diagram that I showed about the reactants, A and is it BC plus BC? That reaction, which was showing free energy and progress of the reaction. Remember, there was a part where there was gives free energy and the part where there was activation energy. So enzymes are very important in that they lower the activation energy which is the energy required to make the substance to react. They lower that. No wonder it becomes faster. However, we're going to explore more on this. So they lower the activation energy. And then enzymes possess or contain active sites. Active sites. So these are sites or places or parts of the enzyme where the reaction takes place. So they're called active sites. So these are the properties of enzymes. Is it clear on the properties of enzymes? Guys, is it clear?
Yes, sir.
Okay, great. Now the enzyme structure, who can try to tell us the enzyme structure? Guys, who can help us with the enzyme structure? Guys, remember we are looking at proteins here. So remember the enzymes, as we have looked at, they contain catalytic properties. They contain catalytic properties. And in addition to that, enzymes will be able to exhibit the physical chemical behavior that are exhibited by proteins. So their solubility, electrolytic behaviors, the chemical reactivity are going to, they are proteins, guys. So they exhibit the properties of the proteins. In short, they're going to have a primary structure, a primary structural configuration. So that configuration and catalytic action of enzyme is really determined by the linear chain of amino acid residues, which are linked by the peptide bonds. And these constitute a protein molecule. And then remember, we have a localized folding of the primary structure. And that's what we call the secondary structure. And then we have the complete folding of the molecule, and that's what we call the tertiary structure. So this is just a revision of the previous lesson. And we said that when you look at the quaternary structure, it's actually the agglomeration of the several folded bonds. Main polypeptides are coming together, forming the quaternary structure. So basically, that's the primary protein structure, which is a sequence of chain of amino acid. This is to contain the acidic carboxylic group. Remember this one, we have amino group, we have the hydrogen atom and the R group or the side chain group. This is generally the structure of a protein enzyme, of an enzyme. So remember I said that these enzymes can form or may not work alone. It would need other substances such as the cofactor. Or it could be a simple, if it's a simple protein, if it's a simple enzyme, it means that it will only contain a protein, just be a protein. So this is a structure of an enzyme. And then we're going to talk more about the prosthetic groups and the core enzymes in our next class, and the upper enzyme as well, how these relate. This is basically the structure of the enzyme. Now, the mechanism of action of enzymes. The mechanism of action of enzymes. So when we look at the mechanism of action, it's actually based on a chemical reaction. Remember, we talked about chemical reaction in the beginning. Chemical, it's based on chemical reaction. So during this chemical reaction, the enzyme is going to bind to the substrate, and this is going to form an enzyme substrate complex, as I will show a picture in a moment. Now, for the mechanism of enzyme action, there are two different models that are postulated or that are put across for the mechanism of enzyme action. So we have the Fisher template model. I'm sure you've heard about this because this is the same as the lock and key model. What did we hear about the lock and key model?
Pardon?
What have we heard about lock and key model in enzymes? Lock and key.
That enzyme will act as the lock, and then the substrate will be the key.
Okay. Thank you very much.
And then
Yes.
there is also an active site.
The
where
what?
the active site.
Okay. Okay. All right. Thank you. So that's to the lock and key theory, the lock and key model. So it's also because of the person who proposed it. So the proposer was Amy Fisher, so now it's called Fisher model. It's simply the same lock and key, famous lock and key theory or model. So when we look at this one, just as he has explained, the model explains what happens, how the interaction happens between a substrate and an enzyme. So this model explains it in terms of a lock and key analog, lock and key. So in this model, we are saying the catalytic site is presumed to be pre-shaped. So the problem with this model, if the catalytic site is presumed to be pre-shaped, it has a drawback. This model has a drawback in that it is implied, or it is implying a rigidity of the active or the catalytic site, in short. There's a rigidity. So as Zia said, the substrate, according to this model, the substrate fits as a key into a lock. The substrate fits as a key into a lock. So in short, we are saying the substrate is the key while the enzyme is the lock. As I said, the drawback of this model is the implied rigidity of the catalytic site. So this model is unable to explain the changes in the structure of the enzyme that occurs in what we are going to consider further as well, what are called allosteric modulators. So we're going to look at allosterics as well. Allosteric enzymes, allosteric modulators. So this model is unable to explain that because of its rigidity in nature. So what happened is that we have the induced fit model. We have the induced fit model. So this was introduced by some man or known as Koshland. So he suggested this model. So it is in contrast to the lock and key model or to the Fisher template model. In contrast to that model, this model suggests a flexible mode for the catalytic site. So he suggested this model. Now the important feature of this is the, as I mentioned, the flexibility of the active site. So what we are saying that in the induced fit model, what happens that when the substrate attaches or when the substrate and the enzymes are attached or are joined, that substrate is going to induce a conformation change. So there will be a conformation change in the active site of the enzyme, so that you're going to find that the substrate is going to fit into the active site in the most convenient way, which is going to promote the chemical reaction. So note the difference. In the Fisher template model, there's rigidity. There's no conformation change that takes place. In the induced fit model, the substrate induces a conformational change in the active site of the enzyme. So this is going to enable the substrate to be able to fit into that active site in the most convenient and efficient way, which promotes the chemical reaction to take place efficiently. Now, this method or model then suggests what is known as competitive inhibition, which we are going to look at on Saturday. It also suggests allosteric modulation, which we're also going to look at. And it also explains what happens during inactivation of enzymes during the denaturization process. The induced fit model suggests competitive inhibition, allosteric modulation, and inactivation of enzymes on denaturization. So these are the mechanism of action of enzymes, the models. Now, this is what we were talking about. We have the active site. This is the enzyme. We have the active site where the reactants are going to fit in a precise position. So that's the active site. Now, when the substrate bind, there is a conformation change in the enzymes, as we have explained in the induced fit model, the induced fit model. So you are saying when ATP and glucose bind to that active site, the enzyme, the shape of the enzyme is going to change. Remember, we talked about the substrate causing or inducing a conformational change. So the conformational change is simply the shape. It's going to change. So this is induced because it is the substrate that have induced that conformational change. So this induced fit is going to reorient the substrate and bind them tighter to the active site, and hence the reaction can take place efficiently. So still under enzyme catalysis, enzyme catalysis can be analyzed as basically a three-step process. Basically a three-step process. So we have the initiation step or phase. So here the reactants are going to bind to the active site in specific orientation. They're going to bind to the active site in a specific orientation. And what is going to be formed is the enzyme substrate complex. I'm going to show summarized version maybe in the next slide to illustrate the process. So the reactants are going to bind to the active site, and what will be formed is an enzyme substrate complex. And then we have the transition state facilitation. Now, in the reaction, in the transition state facilitation, there is an interaction that occurs between the enzyme and the substrate, which lower the activation energy that is required. So that transition state, we are talking about the degree of interaction between the substrate and enzyme. So remember here in the initiation step, there is an interaction between the enzyme and the substrate. Now that degree of interaction that exists between the substrate and the enzyme will increase and reaches a maximum when a temporal unstable intermediate condition. So this is what we call the transition state. It's a temporary unstable intermediate condition is called the transition state. So it is formed during this transition step facilitation. And we are saying that this is going to lower the activation energy that is required. So that's the transition state facilitation. So inside the active site, the catalyst active site, these reactant molecules, they're actually more likely to reach this transition state. Remember, the catalyst is acting as to speed up the reaction. So when these reactant molecules are in the active site, it's easier for them to reach the transition state. So what happens that there is an interaction between the substrate and the R group. Remember the R group, the side chain, the R group in this enzyme's active site, which lowers the required activation energy for the reaction. Thus, this explains why the catalyst is going to proceed much rapidly than when we compare with uncatalyzed reaction. And then there's the termination where the products have a low affinity for active site and hence are released. However, the enzyme is still unchanged even after the reaction. The enzyme is still unchanged even after the reaction. So unlike in the transition state, these products, reaction products, as we have said, they have less affinity. They have less affinity for the active site. So here the binding ends and the enzyme is going to return to its original conformation. Remember there was a change in shape, conformational change. During the termination phase, this is going to reshape to go to its original shape. So don't bother about the name of Michaelis and Menten theorem. Simply, this one simply explains or offers the basis for enzyme mechanism of enzyme action. So this concept, it's a concept of enzyme substrate complex scheme, like we have explained. It's just a summary of what happens. So enzyme reacts, or rather enzyme reacting on the substrate will form the enzyme substrate complex. And then at the termination phase, we'll have the enzyme and the product being released, the enzyme and the product being released. This is simply what is happening. So this is where we come to the end of today's lesson. This is where we come to the end of today's lesson. Any questions?